In this video, we're going to quickly introduce the biological basis for sex determination, or at least the basics of it, as well as look at specific characteristics that are inherited through the sex chromosomes, even if they may have nothing to do with biological sex. So as a little bit of background, remember that all humans with a normal full genome have 23 pairs of chromosomes, the first 22 of which that have nothing to do with biological sex are what we call your autosomes. So autosome is the noun form, so we can refer to chromosomes 1 up to chromosome 22 as your autosomes. If we want to use the adjective form of this word, we would say that these are your autosomal chromosomes, whereas the final two chromosomes here, what we call the X chromosome and the Y chromosome, are simply what we call your sex chromosomes because of the important role that each chromosome plays in biological sex determination, which is the first Punnett square that we're going to be looking at here. So, full disclaimer, biological sex determination has a lot more interesting possibilities that you will learn about as your genetics background becomes more specific, and biological sex determination may or may not influence gender identity. That is a much more complicated question that we will not be speaking of in this video. So, as a full disclaimer, these are the quote-unquote absolute basics of biological sex determination, and most people have the understanding that as long as a person has two X chromosomes, this genotype normally causes an individual to develop as a biological female, while the XY genotype normally causes an individual to develop as a biological male, which leads us to possibly one of the easiest Punnett squares that we can do. Everyone comes from a biological mother and a biological father, regardless of the method of pregnancy, and biological mothers obviously cannot contribute both of their X chromosomes, so they can either give the first one or they can give the second one. Males have all of the responsibility effectively in sex determination because they can either give their X chromosome or their Y chromosome, and this is the critical difference. The top half of our Punnett square would develop as female because both of these individuals have the XX genotype, and therefore this shows that a mother and father have a 50% chance of receiving a biologically female child, whereas the bottom of our Punnett square here represents the 50% chance of developing a biological male child, again, ignoring all of the more interesting situations that involve movement of genes, extra copies of chromosomes, and all of that stuff that you'll learn at higher levels. Now, sex determination is relatively boring because it's relatively easy to predict. What is a lot more interesting for geneticists to explore are what we call sex-linked characteristics whose phenotypes are tied to genotypes on what we call sex-linked genes. Now, a sex-linked gene actually has nothing to do with your sexual characteristics. For example, the condition that we're going to be looking at actually has to do with the functioning of your eyes and the perception of color. Now, because the X chromosome is actually quite large, only a very small percentage of the X chromosome's DNA is specifically related to the biological determination of females. Many other genes are present on the X chromosome as well as on the Y chromosome as well. And one of those conditions that we're going to be looking at here is a condition that we call red-green color blindness, and we can actually do a diagnosis for you to identify whether you are red-green colorblind. Now, I myself am not red-green colorblind, so I can clearly see that in the foreground of this image here, there is a number that is possible to make out in green pigment while the background is in red pigment here. So I can see that this clearly says the number 74. However, if you were red-green colorblind, you have a condition that is actually found on your X chromosome that that renders you unable to tell the difference between red pigments and green pigments. I have no idea what red-green colorblind people would see in this because I'm not red-green colorblind, but what they would not be able to do is distinguish any noticeable difference between the green in the foreground and the red in the background that we see here. This condition is what we call an X-linked genetic condition because the gene that influences red-green color blindness is found on the X chromosome. Now, if a person received a normal X chromosome, such as me, they would be able to make out and perceive two different obvious colors that we would see here, but if they were to have the condition, they would have a different allele that we would actually designate like this. So to designate the presence of the red-green color blindness condition, we would notate an X and usually write a degree sign on it like this in order to indicate the presence of the red-green color blindness condition or any other condition that is X-linked. Now, the good news is for those that are worried about passing this condition on and the difficulties with color identification that it causes, number one, people with red-green color blindness, it's best not to think of it as a disease because they can live perfectly normal and healthy lives. And the second component of that is that the red-green color blindness mutation is actually recessive. So what this means is that if a mother has a normal X chromosome without this mutation, even if they received an X chromosome from the other parent with the mutation, the normal X chromosome would actually override the condition, resulting in this person actually having normal vision. And we'll talk about what the genetic consequences are of this condition here. So now that we have a little bit of background, we can answer a couple of questions here. Firstly, we can answer which sex has a higher chance of being born with red-green color blindness. And what we can do is a rather simple experiment. We can take the hypothetical woman with normal vision here that has one normal X chromosome and one X chromosome with the red-green color blindness mutation and make her, or not make her, but watch as this woman has a voluntary reproductive session with a male with completely normal vision, and we can use the pattern of sex determination in order to track how this X chromosome with the mutation will be inherited. So remember that each parent can only contribute one of their sex chromosomes because of the law of independent assortment, which means if we look at the female offspring or the possibilities for female offspring, we would actually see that both of these females have normal vision because females have two X chromosomes, and as with the mother, the quote-unquote normal X chromosome can actually override the X chromosome that carries the mutation. But something interesting happens with the male children. So if the mother contributes the normal X chromosome to this male child, this male will have normal vision. However, if the male child here receives the X chromosome with the color blindness mutation, now this male child will actually be colorblind. So what we can see from this fairly simple Punnett square that we did is that it is actually male offspring that have a much more likely chance of being born colorblind. Now, because this is because the allele for colorblindness is not just recessive, but specifically X-linked. Now, what this means is that the only situation where a female can experience red-green color blindness is if they end up with a copy of the X chromosome with the color blindness mutation from both of their parents. In other words, only females that are homozygous recessive in the case of the red-green color blindness condition will inherit the condition itself. This daughter here from this particular parentage is what we call a carrier female, because even though this female does not have the red-green color blindness condition because they have a normal X chromosome, what they can still do is pass on this condition to their children, as we see from this male child here. In fact, if this female child, or even if the mother were to have children with a male with red-green color blindness, now it is likely that both male and female children would receive the color blindness condition as well. Now, doing this Punnett square has actually allowed us to calculate what the probability is when a normal male father and a carrier female mother reproduce. The probability of them having a colorblind child, regardless of biological sex, is 25%, or 1/4, because as we've established, it is much more likely that male children will receive the color blindness condition because male children lack a second X chromosome to override the color blindness condition if they receive it from their mother. Now, before you take a look at the practice problems below, whenever dealing with any sex-linked genetic condition, whether they be diseases or not, the very, very first thing that needs to be determined is where the gene is linked to, i.e., is it an X-linked condition where the gene controlling the condition is found on the X chromosome, or whether it is Y-linked and linked to the passing of the Y chromosome, because obviously the inheritance patterns are going to be significantly different. To test your knowledge and double check your skills about not just biological sex determination, but with sex-linked inheritance conditions, you can try the practice problem below, which actually features a weird X-linked dominance condition as opposed to an X-linked recessive condition involving an actual defect with the pigments found in red blood cells. This condition is what we call X-linked protoporphyria, try saying that 10 times fast, which results in the pigments in red blood cells becoming extremely vulnerable to rashes, especially when exposed to light. And the interesting aspect of this condition is that it might actually be biologically tied to myths of vampirism and the idea that vampires were people that had a chronic fear of the sun. But what we can use this condition for is an examination of the inheritance pattern of an X-linked dominant condition distinct from an X-linked recessive condition.